Welcome to AP Biology. Today's presentation is going to be on RNA synthesis. This is a process also known as transcription. So DNA, because it's super important to life and to the success of our cells and our bodies, it stays in the nucleus all the time. It's like a master blueprint of information, and it stays in a super protected safe. The nucleus has two cell membranes around it, so it's about as well protected as you can be inside of a cell. And there are a few types of RNA that we're going to make from our DNA. One is messenger RNA, one is transfer RNA, and one is ribosomal RNA. Messenger RNA is going to be involved in carrying messages from the nucleus out to make proteins. Ribosomal RNA is involved in helping make ribosomes, and transfer RNA is going to be involved in transcription, which is the process of converting the messenger RNA into a protein amino acid sequence. So, because DNA stays in the nucleus, we have to have a molecule that's going to leave the nucleus. And messenger RNA is going to be that molecule. But we want to carry the message from the DNA out to the ribosomes to make the proteins. So in order to do that, we are going to copy the DNA letter for letter with a protein called RNA polymerase. So it works just like DNA polymerase. It's going to copy the letters, and in the process, we're going to make an RNA transcript. And there's some slight differences between DNA and RNA, but the big thing you need to know for RNA transcription is that any place we would have a T, now we have a U. So we have A's with U's, T's with A's, G's with C's, and C's with G's. Okay, but anywhere we would have had a T, now we have a U. So instead of having thymine, we have uracil. But other than that, it pretty much just copies the DNA sequence, and this will be a gene from the beginning to the end of this piece of RNA will be a single gene. In that gene, there will be some exons and some introns, and I'll talk about what we need to do with that in a minute. So RNA has to get processed. It's a rough environment outside of the nucleus, and we have to do a lot of stuff to our RNA to protect it so it lasts long enough that you can actually make proteins. We also have to remove some sequences of it, and that process is called splicing, and we'll talk about that in a minute. So here's our DNA. This is a particular protein. So here's my DNA to make this protein. This is the gene to make this protein. And it's 7,700 base pairs long. And after I translate it, I now have, transcribe it, I now have a primary transcript. That's the initial piece of RNA that comes directly from the DNA. Okay, so it's not what actually leaves to go make proteins, but it's just the initial transcript. You'll notice there's some gold pieces and some green pieces. And most of it's green pieces, believe it or not. And those are the non-coding regions that we have to remove. And then the gold pieces are the coding regions that we have to connect after we remove the non-coding regions. Another thing we have to do to this transcript is we have to throw a poly A tail on, and we have to throw a cap on either end of this. Problem is, when you get out into the cytoplasm, there's a bunch of what we call exonucleases, and they like to chomp messenger RNA starting at the ends and kind of working their way towards the middle. And so what we do is we throw a bunch of extra letters on the ends, and that allows the ends to be chomped without getting into the coding region of the RNA long enough that it can make some protein. So in order to finish off this process of making this primary transcript into our final transcript, we have to remove the introns. That's the turquoise color looking things here. And you have to get down to this final transcript. This is what would be exported into the cytoplasm to actually make our protein. This is what we call a mature messenger RNA. Notice it's quite a bit smaller. So we had 7,700, now we're down to 1,800 base pairs. How do we get rid of these introns? Oh, one easy way to remember what you get rid of and what you keep. Introns stay in the nucleus, and exons, the gold parts, exit the nucleus to go do work. So how do we get rid of the introns? Well, basically you have little proteins that attach to the DNA, and they can find the end of the exon, and then they search along, and then they find the other end of the exon, and then they pull those two ends together, and then so you get like this lariat, they call it, and then it snips here, and it snips here, and it butts those two together, and the lariat is removed. And so you end up with exon 1 connected directly to exon 2, which we connected directly to exon 3, and so on. And then you will have your final messenger RNA that's going to go out and make proteins.